Hi, my name is Andrea, and this summer I spent time in the Nelson lab studying differential gene expression in different species of soybean. So why are soybeans important? They're one of the most agriculturally and economically important crops, as they're a rich source of protein, vitamins, and fiber. So they're very useful in human food or cattle feed. And what's the difference between cultivated and wild soybeans? So wild soybeans have undergone a domestication, while cultivated soybeans have been selectively bred. So they're bred for traits like increased number of nodes, increased flowering time, bigger pods, and bigger roots. And so we're interested in root architecture. So as you can see in this figure here, cultivated soybeans have a lot more lateral roots than the wild species of soybean, Glycine soja. And then next is the root growth rate. So wild soybeans have delayed early root development compared to cultivated, as you can see in this. It's a little hard to see, but there are a lot more lateral roots in the cultivated one versus the wild type. And so for our experiment, this part of the experiment was completed by our collaborators in Pakistan, the Rahman lab. And then over the summer, I did the bioinformatic analysis. So basically, they picked these specific time points, so 3 days of the whole roots, 5 days lateral roots and primary roots, and then at the 7-day mark, they also did lateral roots and primary roots, just to see how different those tissues are. And they prepared them for Illumina sequencing, so RNA extraction. They put all the adapters on, and then they sequenced it. And so we got the raw reads. So what I worked with is first I checked the quality using FastQC. Then I cleaned off the adapter sequences and low quality sequences with Chemomatic. I aligned those clean reads to genome with HiSat2. It's kind of an alignment program. And then we quantified what genes were there in like an assembly transcript with string tie. So next, we wanted to identify the differentially expressed genes. So we used feature counts to actually get the counts of each gene within wild soybean and then cultivated soybean separately, since they're a much different genome. And then, so we paired them together and compared them with the Glycine max as the treatment and then the Glycine soja as a control using DEC2. So it's basically a program that can also do a PCA, kind of check the quality of your sequences, and then give you a whole output spreadsheet of all the differentially expressed genes. And then finally, we looked at those genes. We looked at which ones were highly upregulated or downregulated, and we put them into ShinyGo, which is a genome enrichment program. So for our results, we first have this hierarchical clustering, which is basically demonstrating the quality of the reads and how so each tissue, we did 3 samples each. And then those samples within like the same time point and tissue type, they're very similar to each other, which means there's not much contamination, and they're very easy to compare. And next, we have this Venn diagram here of all the differentially expressed genes, and then specifically, so these are all different conditions, like that's 3 days whole roots, 7 days primary roots, lateral roots, and 5 days primary, 5 days lateral. And basically, this is with Gmax against G soja. So you can see that in the middle, 1,769 genes are shared between all the data sets, and they're conserved throughout the whole root development of the plant. And the ones on the outside here, they're specific to one tissue, which they can still be studied, but for our purposes, we want to focus on the ones that are conserved throughout the whole development. And then 35 of the genes within this middle part are transcription factors, which are especially of interest to us. So finally, we have a gene ontology enrichment analysis. So this basically analyzes genes that you put in, and it shows what pathways those genes are most highly related to. And so what we saw was the genes at the 5-day mark and lateral roots, the genes that are mostly enriched are in fluid transport and water transport, vastly more than the other pathways. So that shows that many of these genes might have a part in root architecture, as the primary role of the roots is to uptake water and search for water. So our conclusions is soybean domestication has had an impact on the genes controlling root development. These shared DEGs may be the conserved genes causing the phenotypic differences in the root system between Glycine max and Glycine soja. And based on genome enrichment, Annotation, genes associated with fluid transport and water transport could be the main cause of the extensive lateral root system in Glycine max during domestication. So for future directions, it will be good to study those 35 DGs that code for transcription factors and perform a functional analysis of those candidate genes associated with extensive root system and Glycine max. And I would like to thank my mentor, Dr. Xiaodan Zhang, Dr. Andrew Nelson, and our collaborator, Dr. Hafiz Rahman. And I would like to thank BTI RU for having us this summer.